Hello, dear students. I welcome you all on behalf of Sri Jendra Rank Zone classes. Dear student, in a previous class of this chapter that is plant breeding, we have initiated a new topic that is tissue culture. And in this tissue culture, what is it? What are the conditions that is required? And in artificially or in a lab, in vitro condition, what type of tissue culture can be carried out and how it is carried out, that all thing we have discussed. Now, in today's second lecture of this tissue culture, we are going to talk about a type of tissue culture. We are going to talk about type of tissue culture based on type of explant involved. Okay, which explant is involved, which type of explant is involved. Based on this, we are going to talk about the type of tissue culture. And in this, the first type of tissue culture we are going to discuss, that is meristem culture. Okay, so if we talk about this meristem culture, dear students, we know that meristem is a region where meristematic tissue are located in the plant, and meristematic tissue are the actively a group of actively dividing cells. Okay, so in this meristem culture, if we talk, this region where these actively dividing cells are present, that we call a apical and axillary meristem. So in meristem culture, apical and axillary meristem, apical and axillary bud or meristem are used as explant. Now the question is why it is used as explant, because they have actively dividing cells, and so during the actively division of these cells, the pathogen cannot infect this particular cells. So now they have this meristem, this meristem are free of virus due to high concentration of high concentration of oxygen and rapid rate of cell division. So due to rapid rate of division and high concentration of oxygen, the pathogen or virus cannot infect it, and so because of this, these meristem are free of viruses. So this meristem are used to create or obtain pathogen free pathogen or virus free plant. So if we want to create a virus free or pathogen free plant, then we will select a explant as a apical or axillary bud. So for this, what it is done? So for this, in the process, the apical meristem with one or two leaf primordia. Leaf primordia means initial leaf, are taken as explant. Okay, as explant. And for this, the apical bud is sterilized. Then this apical meristem with the two leaf primordia are taken, which is also known as the shoot tip. So now after this shoot, after sterilization, shoot tip is placed over culture medium under aseptic or sterile condition. So aseptic or sterile condition are the virus-free conditions, and the plant will develop. So such a way the meristem culture can be done. And it is known that several scientists, if we talk, several scientists have succeeded in culturing meristems of banana, sugarcane, and potato. Okay, so what did the scientists have been succeeded? Okay, so this is what a first type of culture, that is meristem culture. After this, we are going to talk about the second type of culture, that is known as embryo culture. If we talk about embryo culture, in this, first, what it is done to carry out this embryo culture, excision or removal of young embryo from from seeds, and then their cultivation through tissue culture. Their cultivation through tissue culture. So it is excision or removal of young embryo from the seeds and their cultivation through the tissue culture. Now, if we talk about this embryo culture, it has certain application. And if we talk about certain application, what is here in embryo culture, what we need to do? Just remove the embryo from the seed and put it over the culture bed, and it will develop. And through tissue culture, it will develop and give a new plant. So, what are the application? So, in application, the first application is the interspecific hybrids are often sterile. It is known that interspecific hybrid are often sterile, means they are not able to give a progeny because of embryo mortality, means the death of embryo. And another reason is seed collapse. These two things happen in the interspecific hybrid, so they cannot give a next offspring. So in this, embryo rescue can be done. And to carry out this embryo rescue, what can be done? To carry out this embryo rescue, the hybrid embryo is excised or removed from female parent in early stage. In early stage, and then it is cultured, so the embryo will form a new plantlet. So such a way, the particularly embryo rescue can be carried out in the interspecific hybrid. And if we take an example of this, Then the hybrid between common bean, whose scientific name is Phaseolus vulgaris, that and wild bean, and this wild bean is Phaseolus angustissimus. Angustissimus. Okay, so this between the hybrid can be cultured through the embryo culture. The another application of this is embryo culture allow seedlings development in plants whose seeds whose seeds lack stored nutrients required for seedling growth. So if we take here example is orchid. Okay, so in orchid, if there is a nutrient are not there, in that case we can remove the embryo and can go and culture on the plates. Then it can help us for the what in particularly growth of the seedlings. And it's also embryo culture is also used in multiplication of some rare plant like macapuno coconut. Macapuno coconuts. Okay, so in that they can be used. After this embryo culture, the third type of culture that can be done based on the explant, that is haploid culture or androgenic haploid culture, or we can say pollen grain culture. So this thing, that is haploid culture, androgenic haploid culture or pollen grain culture, is done here, and this technique was developed by this pollen grain culture technique. This technique developed by Guha and Maheshwari. And Guha and Maheshwari are the scientists who developed this technique in Datura, Inoxia. Okay, and here how this is done. So first, so here first floral bud are taken, which are very young and unopened. Okay, these floral buds are taken, and then this floral bud are sterilized in Clorox, that is antimicrobial chemical for 20 to 40 minutes. So the surface sterilization will be done and then the floral bud will be free of the microbes. Now then, floral bud are opened. Floral bud are opened to remove. Floral bud are opened to remove anthers. Okay. Then these anthers are introduced over culture medium. So these anthers are introduced over a culture medium, and it is known that within 4 to 6 weeks, each anther give rise to a number of haploid embryoids. So such a way, this number of haploids will develop from each pollen grain of the anther. So number of haploids embryoids will develop from the one anther or each anther. Now, normally they produce sterile haploid plants. So sterile haploid plants are developed by this. Now, why? Because they are the haploid plant. So if we want to from this a diploid plant, so it is known that colchicine is a one chemical and it is a one type of mitotic poison which carry out polyploidy or increase in set. So it is known that colchicine treatment of the cells results in chromosome doubling and produces homozygous diploid for each and every trait. For each and every trait. And it is known that gynogenic haploids are also possible or produced by unfertilized ovules. Means if unfertilized ovules are developed like the anther or pollens, then they also can give rise to the haploid embryoids. And now it is known that winter wheat variety like Jingua 1 and rice Kuan 18 are two important variety two important varieties produced by this technique. Okay, and if we talk about use of this haploid culture, then uses of androgenic haploid, if we talk, then the first use is useful in mutational breeding. Useful in mutation breeding. Second is to maintain pure line or homozygous line. And third, to produce seedless variety. So such a way the androgenic haploid can be useful. Okay, so this is the third type of haploid culture, or we can say androgenic culture, which we have discussed. Now, after this, the fourth type of culture which we are going to discuss, that is protoplast fusion or somatic hybridization. Protoplast fusion or somatic hybridization that we are going to discuss.